Fine. Students, so we will look into the nitrogenous compounds which are important in the fermentation medium. Okay, one of them is corn chip liquor, where the corn starch is produced with gluten and other corn products we produce. No, that remaining water is called as corn chip liquor. Next is soybean meal. Soybeans are used for the various like extractions, and the remaining pulp, whatever is left out, is called a soybean meal, which is having a lot of nitrogen source. Parva media is another thing. Parva media means the cotton seeds, no? Those are taken and powdered. That is having a lot of protein. 56% of protein will be present, and 24% of carbohydrates will be there. Some oils, 5%, and ash, like ions. I mean to calcium, iron, phosphorus, chlorine. Okay, this are present in the parva media. And distillery solubles can also be used. Okay, distillery soluble means after the alcohol production is done, the remaining grain mash is there, no, that can be also used as a source of nitrogen. Okay, which is mainly it's concentrated and dried. They get the dried form of the grains, that is used in the production as a nitrogen source. Oils and fats are very important in the media while adding. Because oils like vegetable oils like olives oil, maize oil, cottonseed oil, linseed oil, which is a flax, I mean to say, soybean oil, they're all used as anti-foaming agents. So even if you add soybean meal, it's indirectly you're giving a anti-foaming agent, natural anti-foaming agent. Okay, nitrogen sources can also include the inorganic nitrogen like ammonia gas. Okay, which is a very important thing which can control the pH also. Ammonium salts, like you can maintain the acidic conditions in the medium, and nitrites also can be used. It is once the ammonium salts get exhausted, we can go for nitrates also, which is helpful in regulating the pH. Organic nitrogen can be used like a protein source, urea. Natural proteins like corn chip, like soybean meal, corn chip liquor, peanut meal, yeast extract, cottonseed oil, such things can be used as an mainly for organic nitrogen sources. Of course, there are minerals required. So we all know that magnesium, phosphorus, potassium, sulfur, calcium, copper, cobalt, iron, manganese, molybdenum, zinc, all these are required. We give them in the form of a combined form like potassium dihydrogen phosphate, zinc sulfate, ferrous sulfate, etc. Right? These all are added components in the fermentation medium, other than the normal major elements. These are called as minor elements what you are providing in the fermentation medium. So these all are going to be part of the media formulation. Chelators are required. Okay, chelators are very important because the visible precipitate of the insoluble metals are in media. Okay, insoluble metals, visible precipitates will be present, so that and add a lower concentration of chelating agents that time. Like EDTA can be used, citric acid, which is a very natural citric acid, can be used, yeast extract, okay, peptones, all these can be used as chelating agents. And coming to growth factors, vitamins, amino acids, fatty acids. So I told you these trace elements and all, no, growth factors and all, they are very much required for the enzyme activities. Okay, it may act as enzyme activators like cofactors, right? Okay, mainly they're used like calcium pantothenate, biotin. These are some of the growth factors. Buffers are also going to be added, like calcium carbonate, maintains pH 7, then phosphates, then proteins, peptides, amino acids, corn chip liquor, they all act as buffering agents. And precursors, sometimes we may have to add precursors in the medium, like for example phenylethylamine is added, or phenylacetic acid, a part of phenylacetic acid is used, which contains this component phenylethylamine, and then for the penicillin production. Inhibitors, some of them are also like inhibitors. During ethanol fermentation, they add sodium bisulfite. Why? Because it can convert acetaldehyde to glycerol if you're going for glycerol manufacturing. Okay, so there's a deviation. Sometimes we may have to use inhibitors for the production of, otherwise they would have been producing acetaldehyde to alcohol. You're deviating it from acetaldehyde to glycerol, like that. Inducers, Okay, sometimes inducers are added, like as I told you earlier, maltodextrins for inducing amylase, fatty acids like for inducing lipase, starch for inducing amylase, like that. So sometimes may have to add inducers for specific productions, right? So hence I've come back to the first slide, saying you that the media formulation plays a very important role in the industry, because uh, one has to think about the carbon source, nitrogen source, how much of aeration to be given. If you give these many hours of aeration and this much of carbon source, all it's all calculative, okay, and this much biomass is produced. So it's they all have to be like standardized before it goes for the major pilot plant productions. So in the lab scales, they have to, every step they have to see that this basic stoichiometry equations are strictly followed, and what are the important metabolites being produced. So like sufficient screening is done and standardization is done, only then they have to execute it in the form of a production. Okay, so I think with some basic idea of what is media, how to formulate media, how to enhance the production, how to minimize the unwanted substances, I mean unwanted metabolites, what type of starch media is required, or from molasses are required, or can we use starch hydrolysis? I was asking a question that time. Can we use starch hydrolysates for production of alcohol? What's your answer for this? Yes or no? Okay, it should be yes, because we can use even potatoes for production of alcohol by using yeast as an inoculum. How? We can use a starch hydrolysate of potato. It will convert to sugars, right? Then you can inoculate the yeast. We all know that yeast cannot break down the starch. It can't produce amylase, right? So yeast can use the sugars which are released from the starch. How can you execute it? By boiling, then by adding acids, by adding amylase. Of course, you'll add amylase after the media is cooled. Okay, that's all the procedure. So different steps you can follow, and you can produce the simple sugars so that yeast can directly use them for the utilization. That's how even in beer production, beer malt is barley malt is there, isn't it? Barley malt only is having starch. You're cooking, you're breaking down the starch, you're adding after this actually you're adding amylase also in the beer production. Amylase will break down the starch to sugars, right? Yeah. So we all know that means starchy grains, anything you can convert to simple glucose and use yeast for alcohol production. With such simple examples, one has to understand that media formulation plays a very important role in the industry. Without media formulation, we cannot, without crude media formulation, standardization, we cannot run the industry. And this becomes mandatory, because we have to see the nutritive value of the raw material, we have to see the quality control of the microbes in the raw material, whatever comes into the industry, and then they should go for some processing will be there. Let's say grains are there, storage of the grains should be done properly, there should not be any pests, so pest controls standards have to be maintained. Then you have to go for storage of these grains, and then you have to go for the quality checks, and then the preparation. So a lot of things come. It's not a easy task, right? So all that, every batch, they have to do the scale up, every batch before it goes for the main production. Every batch has to be standardized. It has to follow up. It has to follow the media formulations, regulations. A lot of things are there. It's not a simple task. So industry is always like, on a task of completing them, keep on monitoring them. It's a very continuous process. If they cannot neglect any step. Okay, every step is important to standardize, so that you are satisfying this formula. It's only a formula written here. So there's a lot of instrumentations, bioinstrumentations. They have to use instruments to see how much carbon source is there, how much nitrogen source is there. A lot of assays have to be done. Okay, so it's a huge task. Okay, we're only discussing the outline, I can say. If you really understand every step, it's such a tough task. You have to see biomass, how much biomass is there. Let's say, I'll tell you a simple example for this. Let's say you use a groundnut powder for nitrogen source. Let's say you use a rice flour here. I'm telling an example. You can formulate this. You get the growth, means you will use a culture flask in the lab to grow. After that, what you will do, you will filter the conical flask containing the microbe. You will find out the wet weight, dry weight, which is doing in the laboratories many a times. In the first semester, second semester, no, you're done. No, first semester, fungal biomass and all, right, like that, measuring that one. Then whatever metabolites they're producing, whether that's having antimicrobial, whether it's having some enhancers, lots and lots. Time of incubation matters. If you leave it for 15 hours, the metabolite is different. If you leave it for 20 hours, metabolite is different. Correct? So every task has to, the heat which is generated, whether it will affect the metabolite. It's a connectivity. It's a very tough part, but very challenging. Okay, media formulation is very challenging. One has to really explore that. You will understand the importance of that in the industry. Once you explore it, once you understand it, once you give a better outcome, the industry will appreciate that person, appreciate the team which is involved in this process. Okay, so this basic talk, maybe you would have come to a conclusion what is actually media formulation. Okay, if you have any queries, we will again get back to you and answer them. Okay, students, with this basic informations what we have shared, we will for today's session will end up, but we will meet with a few more topics on industrial microbiology basics, and we'll meet for course chat. Okay, I'll inform you. We'll meet in the WhatsApp. We'll inform you for the course chat. We'll be discussing any queries in the course chat shortly. Thank you.